Welcome everybody to the first session of the Ancient Genomics seminar series of this term. So we are starting a very great course of talks with Cosimo Posth. Cosimo is working on ancient DNA, so it's really the core topic of the seminar series. He did his PhD in Tübingen, was working as first a postdoc and then PI at the Max Planck Institute in Jena, when it was still in Jena. And since 2020, he has been a junior professor in Tübingen, and he has worked on various topics in ancient human history, recent human history, in different parts of the world, basically from Neanderthals to recent populations. And yeah, so we're looking forward to what you have to tell us about the hunter-gatherers of Europe.
Yes, thanks, Martin. Thanks for the invitation. Thanks, everybody, for coming here. It's actually an honor to be here. I was really hoping to be invited once, and finally I got invited. Great place, great people. So thanks a lot for having me today, telling you a bit about archaeogenomics of European hunter-gatherers. I thought that this topic might interest many of you guys, so I decided to present on this. There will be some published stuff, some unpublished stuff, so I'm happy to share with you. So let's get started. The topic I would like to cover today are four. Mention and start a bit the topic about setting the stage of how is the genetic profile of these so-called initial Europeans. I have to stress again that the topic of my talk will not be focusing on Neanderthals. So when talking initial Europeans, I'm referring to early modern humans. Then moving on to Upper Paleolithic to Neolithic hunter-gatherers, this large data set that we have assembled to see the genomic transformation over more than 30,000 years. We'll present a recently accepted study focusing on interdisciplinary analysis of Riparo Tagliente, so epigravettian human remains, where we perform a biomolecular study integrating multiple levels of evidence. And then, last but not least, I would like to introduce you to our newly founded Leibniz Science Campus. It's called GACT, Genomic Archaeology Campus Tübingen, which mainly focus on the analysis on ancient DNA from sediment. And here I know that many of you guys are working on similar topics. Should I click OK? Yes, okay, good. So here, we cannot start with this slide here. Many in the rooms here look specific to Ron, but many others have contributed to this increased boost of ancient DNA genome being published until now. Here at the beginning of, I think this was the beginning of 2023, when this was submitted, there were 10,000 genome or ancient individuals with genome-wide data being available. Now, probably we are already in 1 or 2 years after reaching the 20,000 genomes, and the number is growing. Of course, you see a huge discrepancy, a huge distribution unevenness of these ancient genomes, both in this case geographic unevenness, with certain areas that have been much more studied than others because of multiple reasons, ethical reasons, archaeological tradition, preservation issue, and so forth. But if we were able to focus on Europe specifically and focus then on the temporal frame, here we see the geographical frame, but the temporal frame, this is another picture. So this is again from 2020, a figure from a review I wrote with Inigo Lalde. Of course, it's super outdated after 4 years, but it gives you the general trend. Here, time is from left is younger and right is older on the x-axis. On the y-axis, number of individuals with genome-wide data, and divided by Europe and Near East in reddish and greenish. And you see that there is a huge amount of ancient DNA data generated from 8,000 years onwards, and then again a big peak around 5,000 years. And this is really because early studies focusing on ancient genomic data were interested to capture possible genetic transformation associated with major cultural transformation. For example, at the beginning, of course, around 8,000 years ago, the advent of agriculture, the Neolithization, and then 5,000 years ago, the advent soon after of the Bronze Age. But if you really see what is older than 8,500 is extremely limited data, okay? Despite, obviously, humans were there, modern humans were there for several tens of thousands of years. So in this talk, I would like to focus specifically on what we say Ice Age Europe, specifically focusing on ancient DNA retrieval from European hunter-gatherers, and focusing on the genomic transformation from Upper Paleolithic to the Late Neolithic. So in a time period between 45,000 and 5,000 years ago. We're talking about 40,000 years of human history. You see here on this arrow is a huge amount of data, and of time, and not so little data. And it goes all until basically the end of the Neolithic, because there were still people relying on a foraging lifestyle so late in time in certain areas of Europe, for example, the northern fringes of Europe. And so what's the goal of this kind of research that I've been conducting in the past years is, first of all, to try to see if what is the role of previously proposed climatic events in the demographic history or demographic history of these hunter-gatherers, and also assess if genetic similarity correspond to associated archaeological culture across Europe, okay? So when we start with this framework, again, I think we really need to start with the initial Europeans. Who are those? There are few skeletons being found, but there are even fewer genome or genome-wide data being generated. And here, those are the so-called VIPs of the Upper Paleolithic. At least I like to call them like that. So there is Oase. Many of you know them by name. Oase 1, this mandible from Romania. Bacciocchiro, unfortunately, the picture really disappeared, but there are few bones and one tooth actually from the initial Upper Paleolithic from Bulgaria. Zlatý, I will discuss about this very important specimen in a few slides. And then most recently, the data generated so far, only mitochondrial DNA data from Ranis, associated with LRJ culture from Germany. But in this period, these early initial Europeans had to coexist with other forms of humans. We all know about the presence of Neanderthals, who, based on this, very pivotal study by Tom in 2014, were there at least until 40,000 years ago. So this gives us at least 5,000 years of geographical and temporal overlap between Neanderthals and early modern humans. So the big question is, how do these initial Europeans look like? Do they have extra Neanderthal ancestry? Do they have the same? Are they related to later population that we find in Europe? And so I'd like to start briefly with Oase 1, this mandible. In 2015, Eric Trinkaus, sorry, in 2003, Eric Trinkaus described that as a hybrid. So based on the morphology of the mandible, thought that there were some mixed traits morphologically, both resembling modern humans and Neanderthals. Of course, it was very highly contested, this, because based on morphology, it's very hard to actually be certain about that. But it's a very interesting site because you see it's located within the known distribution of Neanderthals in blue. And so Pestera Oase as well as Bacciocchiro Cave are both located in the Balkans in an area where we know Neanderthals were present at a certain point in their history, right? And in 2015, when Xiaomei-Fu published their reconstruction of the genome, and then in 2021, where Mateja Ajanak published the Bacciocchiro data, turned out that two major findings. First of all, that both these populations had an extra Neanderthal contribution, so much more than present-day people, up to 10% of Neanderthal ancestry. And based on the length of these Neanderthal blocks in their genome, turned out that they had a Neanderthal ancestor as close in their family trees as 4 generations. So really the idea of hybrids, as it was proposed by Brinkhaus in 2003, was not actually to be dismissed. These are really individuals, and most likely population, which had recent ancestors in their family tree deriving from Neanderthal. So very on top of the ancestry of Neanderthal ancestry, all non-Africans share, they have an additional contribution, which most likely happen within Europe as they expand within the continent. So I had the chance to actually to work together with some people in the room here on this very special fossil, which I would tell you the story because I think it's a peculiar story, and I even wrote a blog about that because it was one of the most surprising findings in my career. We got access to skull, almost complete skull from the regions around Prague in Bohemia. It's called Zlatý in Czechia, and the context is very uncertain. So archaeologically, it's not been associated with any lithic industry. It's kind of a skull found in a cave without context, right? And just want to show you here how well it's preserved. We, of course, 3D scans before sampling it. And you see, okay, one part is largely missing, but you know, you have the mandible entirely preserved and large part of also the cranium. And so it's a very unique level of preservation. So together with Jaroslav Bruzek and Petr Veleminski, colleagues from Prague, they came to Jena and we did the sampling of this specimen targeting the petrous bone as taught us, very deep in the densest area to really obtain 15 milligrams of bone powder, from which we then perform genetic analysis. Of course, the goal is to understand how old the individual is, but also how does this Paleolithic individual relates to other hunter-gatherers from Europe. Maybe you don't notice here in the picture, but just to give you a hint here, if you look at the mandible on the right-hand side, you see some shining on the mandible, some shining, color, and this is kind of a hint of what's going to happen after. So first of all, we usually do mitochondrial DNA analysis as is a standard approach to see what is the preservation and to reconstruct the phylogeny. And here is what happens. So we reconstruct the mitochondrial genome of Zlatý, we place it on a phylogeny, and it places on the N lineage, so as many other Europeans, but extremely basal. And if you go and check how many substitutions this Zlatý has from the node of the N lineage, is only 3 substitutions. And in comparison, Bashokiro, which were directly dated in Hablan et al. 2020 to 47 to 45,000 years, they have even 1 extra substitution compared to Zlatý. So we were very surprised to see, okay, we're probably dealing with something extremely old, which has even less substitution than something that is dated to around 45,000 years, okay? And as you know, I mean, mutations and substitution accumulates over time. So the fewer mutation they have, the older you're supposed to be, right? So we did beast analysis just to do with a molecular clock and see in which ballpark age this will land. And we obtain a date of 43,000. But that's why now Katerina and Tom are happy. Our dates are not as precise as their radiocarbon dates. So we have a huge high posterior density interval between 31 and 53,000. Basically means nothing. But at least it's something to tell us it's very old, right? We have really to try to date the specimen again, try to see if in which ballpark the genome is. So, and here comes the painful story. This is the date when we got, no, we didn't get, this is the date that was already available from 2002. When we got the samples, they told us, okay, this specimen is supposed to be Magdalenian based on their date. So around, 16,000 calibrated BP. So we first, when we got these results, we sent it to Mannheim, we tried to date it there, and we got 28,000. So already completely not overlapping, much older, but still not old enough, right? So we said, okay, if we want the oldest date, we send it to Tom. And look at that. No, it became younger. So again, not overlapping. Then we said, okay, Tom said, okay, no worry, I have a method, I have hydroxyproline dating that's gonna nail it, okay? And in fact, we have to be honest, he got the oldest date, yes, 34,000 years ago, but still, this was not consistent with our expectation based on the molecular dating, which we expect to be older than 40,000. So something must have happened. So we submitted the paper to, I think we was submitted first to Nature, rejected, Science straight out, Cell revision, rejected. So we were like, okay, man, nobody believed that. They want the direct date, right? They really want to say, okay, tell me the date is 35,000. Is the oldest that you have? We believe that. And we said, look, it's older than that. And so we decided to look actually in the non-human DNA that we had extracted deep inside from the petrous bones, okay? Imagine like deep inside the petrous bone. And so we look at the non-human DNA. There was around 10% of human DNA, all the 90% rest. We did metagenomic analysis, and we were actually able to reconstruct a full mitochondrial genome just based on the shotgun data from a cow. And we were also able to put this cow mitochondrial DNA on a phylogeny, and turned out it's a European cow on the T3 haplogroups, okay? So we put the pieces of the puzzle together based on analysis that Tom had done in his lab, show evidence of contamination based on the glowing and the shininess of the bones. And we kind of reconstructed and said that what we think it happens is that it was actually collagen, cattle collagen glue, so collagen made of, glue made of cattle collagen that was used as a consolidant, which most likely penetrated deep into the skull, prevented us to have a reliable radical bond date. Even hydroxyproline was able to remove some of the other contamination, but was not able to remove, of course, collagen contamination, right? So we went on and say, okay, let's try to use other methods to date these important genomes, because it looks to be very old. And we went back and checked the nuclear genome, not only the mitochondrial DNA, but the nuclear genome, and said, okay, first of all, let's see what is the percentage of Neanderthal ancestry. And here you see on the left-hand side that Zlatý has around 3% of Neanderthal ancestry, not significantly higher than other Upper Paleolithic Europeans, okay? So it doesn't seem to be like Bachokiro or Oase with extra Neanderthal mixture. And then we looked at the length of these Neanderthal blocks in the genome of Zlatý. This was a shotgun genome of around 2x. And we could estimate that this individual lived 60 to 80 generations before the initial Neanderthal mixture. Okay, so it's not a hybrid individual, okay, but this individual lived maybe 2,000, 3,000 years before there was the Neanderthal mixture, the major Neanderthal mixture into the modern human population. And based on previously dated modern humans, for example, Ustishi, for which we have the genome and we have a reliable radical bond date, we could assess and estimate that based on the Neanderthal length, we have this genome or this individual which is supposed to be dated older than 45,000 years. But of course, older than 45,000 is a very relative dates, but we couldn't say more than that. We could say, based on what we know from other genomes that are reliably dating and looking at the length, this individual even has larger blocks of the Neanderthal ancestry than Ustishi, meaning that this individual is even closer in time to the Neanderthal admixture, meaning that is even older than Ustishi. So it has to be older than 45,000. How old? Who knows? So we published this study in 2021. At the end, came out in Nature Ecology Evolution. We are still happy to get it out. Oldest modern human skull from Europe. And also something very interesting I didn't mention, this individual branches off from non-Africans before European and Asian divergence. So imagine, Europeans and Asians, this is also another indication of its antiquity. European and Asians diverge at least 40,000, right? This individual is more basal than this split, right? Is an outgroup to both European and Asian, okay? And also very interesting, we see that this Zlatý genome and also other pre-40,000 years old humans largely disappears from Europe. And this is also something similar that happened to Neanderthals, as you know, 40,000. So it seems that this, at least based on the limited knowledge that we have so far on the initial Upper Paleolithic or early Europeans that we find they genetically do not contribute substantially to later European population. So something must have happened. And many of these early offshoots of modern human lineages which reached Europe before 40,000 did not make it afterward. So I would like now to move to the part where we actually know that there is some genetic contribution to later population, and this is starting from after 40,000 years. And in 2016, we published a study with 50 hunter-gatherer genome from all across Europe, spanning a large time frame between the origination all the way to Mesolithic and even Neolithic. And you see here that, yes, there was large temporal distribution, but you can imagine with 50 genomes for 40,000 years of human history, it's basically nothing. So there were huge amount of gaps in our understanding what is genetic continuity between different periods. What happens during the last glacial maximum? Is there, let's say, synchronic or diachronic connections, genetic connection? And here you can see kind of a schematic representation of the different chronocultural subdivision of the Upper Paleolithic, Aurignacian, Gravettian, and so forth, all the way to the Mesolithic. And of course, this is a simplification of how geographically is distributed material culture and lithic technology across Europe. But it's just for us to have a bit of a reference where we are in time and space. So in this study published last year, we generated 116 new individuals from 54 sites across Europe and also one site in Central Asia. And this is really basically 1/3 of all hunter-gatherer genome being published. In total, is 360 hunter-gatherer genomes available. And if you see our new data where it fits is between 35 and 5,000 years. This is the new data spanning before the last glacial maximum, after the last glacial maximum, many also from the hunter-gatherer from the Holocene period. But importantly, we got access also to 2 individuals during the last glacial maximum. So really a period where we know that there was a contraction of population. It's very hard to find human specimens and also to obtain DNA from those. So we started from the Gravettian period where we had more data. And Gravettian is an archaeological culture or lithic technology, or as you name it, which is known to be pan-European. It's really like spread in many, many regions of Europe. And there was a big question: is there genetic homogeneity or heterogeneity among this Gravettian population in a time frame between 35 and 26,000 years before present? And so here in this MDS plot on the right, you can see that once you exclude the pre-40,000 individuals, we group all together, not because they're more closely related to each other, just because they are equally distant from all the rest, okay? So they form a cluster, but it doesn't represent a real cluster of genetic similarities. All these lineages are equally distant from the rest, but they are not really particularly close to each other. But then if you focus on after the 40,000, you see 2 clusters, right? You see what we call the Vestonice cluster, represented by East and South Gravettian individuals from Czech Republic, Austria, but also from Southern Italy. And then another cluster of so-called West Gravettian, right? The people from Spain, Southwestern France. And if we compare and see where the previously published Aurignacian genomes fall, we see that this Aurignacian genome from Belgium, Goyer Q016, actually falls very similarly genetically to West Gravettian. So there is some sort of genetic continuity between the Aurignacian we have available so far and West Gravettian population, okay? But what is important, there is a big divide, 2 clear distinct clusters between West Gravettian and East/South Gravettian, okay? Contrary to the expectation of a more homogeneous population because of material culture, there is of course evidence of a lot of connection between these groups. If you move after the Solutrean period, and here we have, of course, very limited data, but the Solutrean is a archaeological culture that is during the LGM, okay, between 25 and 19,000 years ago in southwestern Europe, so Iberia as well as southwestern France. And here we again do the same MDS plot. And what you see here is that, maybe I have an arrow. Yes, the Solutrean associated individual fall both temporally, but also genetically intermediate between West Gravetian, that I mentioned before, and Magdalenian. Magdalenian are the population that come, or at least is the culture that comes after the LGM. And so there seems to be a continuum and some sort of genetic continuity in Southwestern Europe during the LGM. And this is really reinforcing the idea of Iberia, as well as Southwestern France, as a potential glacial refugium for human populations during the LGM. So there is indeed genetic continuity through this harsh and coldest period of the last ice age. Moving on into the later time period, so moving on into the post-LGM, you see that Europe, there are two major archaeological cultures, the Magdalenian associated individuals from South and Southwestern to Eastern Europe, as well as the Epigravetian associated individual from Southern Europe and Southeastern Europe, the Balkans and further east. And so here we were able to generate data in red on new individuals from associated Magdalenian culture, both from the western end and as well to the eastern end, all the way to Poland, so Maciskaya site, associated with Magdalenian culture. And what we see here is that by doing an ancestry modeling, these individuals are very homogeneous, okay? So it seems that these Magdalenians, excluding Elmiron, which is a bit an exception, but these Magdalenians from Central Europe, Western Europe, all the way to Eastern Europe, they most likely derive from common source populations. And from this source population, there was a re-expansion after the LGM all the way to Eastern Europe. And now we know actually even to the British Isle, okay? So imagine this place was not inhabitable during the LGM, but after the LGM, there was most likely a southwest to northeast population expansion of a population, Magdalenian associated population, which have very similar genetic profile and spread all over the place, okay? In Iberia, again, is a bit of a different story, but we see a homogeneous pattern in the rest of Europe, or at least Northern Europe. Instead, in Southern Europe, there is, as I mentioned, a different culture, the Epigravetian, still during the LGM. And here we generated data not only from Southern Italy in Sicily, but also mainly from Northern Italy, new genomes analyzed with previously published data. And here, again, my favorite analysis, MDS plot for the Upper Paleolithic. You see that the Italian Epigravetian are extremely homogeneous, again, like the Magdalenian. But what we find very remarkable is that if you compare this Epigravetian to the Italian Gravetian, as you know, the Gravetian are the one that were there before, from South Italy, there is no evidence of genetic continuity. So we couldn't find any evidence of genetic contribution between the Gravetian and the Epigravetian in the same geographic location. So discontinuity between pre and post-LGM Italian hunter-gatherers, and this really questions the role of the Italian peninsula as climatic refugium during the LGM. Okay, so we see that Iberia and southwestern France is a putative region for a refugium for modern humans during the LGM. Italy, based on our analysis, we don't have so much data, but doesn't seem to be really a place with direct genetic continuity between the Gravetian and the Epigravetian. So something must have happened genetically, a major replacement between these phases. Even more interesting, and I want to spend a second on this slide because we have new published data on this, is how are those Epigravetian individuals related to each other? So we try to perform, kind of reconstruct a tree based on neighbor joining tree based on F2 statistics, basically is a measure of genetic distance between single individuals. And we could actually reconstruct almost like a purse on a chain, like a tree that has a very strong phylogeographic pattern. So there is a strong spatial and non-temporal structure. What does it mean? It means that genomes from northern Italy seems to be closer to each other, from central Italy closer to each other, and southern Italy closer to each other, independent of their age, okay? And so we could kind of propose an idea that this epigravettian ancestry arrived into Europe, most likely from the east, based on other evidence, but most likely from the Balkans, and entered towards the north of Italy and start splitting into different lineages and moving gradually towards the south of Italy, diverging and drifting off as they move from north to south. And we know about this drifting off because we can measure actually genetic diversity. And if you compare these three groups, group 1, group 2, and group 3, moving from north, central, and south of Italy, we observe a gradual lower diversity, so decrease in heterozygosity, moving south along the Italian peninsula, okay? So you arrive in Sicily that you have individuals that are so closely related to each other, they almost look cousins, but they're not family related. It's just that they went through subsequent bottlenecks, moving from the north to the south, reaching the Sicily, without any evidence of admixtures along the way. We don't see that these individuals actually have in the south a higher Gravettian-like ancestry. It seems that this movement replaced the previous inhabitants of the Italian peninsula. And last week, I believe, or maybe 2 weeks ago, we actually published, together with a team in Florence and in Ravenna, a genome from Lemura. I don't know if you had the time to actually see this publication. Is an infant burial from southern Italy in Puglia, again, dated to 17,000 years ago. And if we do a similar analysis, in this case it's called a QP graph, but it's very similar analysis to this neighbor joining tree that I presented before, Lemura individual is branching off together with other southern Italians, in this case Sicilian, okay? What does it mean? What are the implications? First of all, we confirm this strong phylogeographic pattern, okay? So people that are geographically more closely related to each other, independence of their dates. Second, it has a very important implication because you imagine that if Lemura is 17,000 years ago, okay, it means that arrival of this ancestry in Italy needs to be much before 17,000, right? If Lemura is 17,000 is so much derived, the arrival of this lineage with Tagliente, but even before with other more deeply divergent lineage of the Epigravettian needs to arrive before. And so we proposed in the paper back then, and also in the study, that probably this turnover event didn't happen within the Epigravettian, but happened probably at the transition between Gravettian and Epigravettian, most likely during the LGM, you know? So again, reinforce the idea that Italy maybe was not the best place to be during the LGM, contrary to the expectation of always warm and nice weather. Okay, and then I'm gonna go to the final part of this time frame, final Paleolithic and Mesolithic, and here you know that in the ancient DNA communities, these are called usually the Western hunter-gatherers, WHG. And what we find again that all post 14,000 individuals from Central Europe have this Epigravettian-like ancestry. We can model that more formally, not only with PCA, but with QPADM, and see that actually, starting with Oberkassel is dated to 14,000 years ago, all the way down to 7,000 years ago. These individuals are extremely homogeneous, okay? So they're carrying an ancestry, that originally comes from Italy, Epigravettian-like ancestry, mix only a bit with local population, this kind of more Magdalenian-like population, and then spread all over the place, okay? But there is, again, a large-scale replacement of the previous population, the Magdalenian-associated individuals, with this ancestry comes from the south, okay? So it seems that the Epigravettian-like ancestry, which first replaced the Gravettian in Italy, few thousand years after, spread north of the Alps and replaced the Magdalenian ancestry in Central Europe, okay? And you see very strong continuity, starting with Oberkassel, which is the old individual from Central Europe with his ancestry. We label this ancestry just to make it more complicated, Oberkassel. Nobody liked it. Everybody prefers to still use the name Western hunter-gatherers, which whatever it means, Western of what. But in any case, we got at least a very nice article on the Frankfurter Allgemeine. Oberkassel ist überall. So they were very happy, the Germans. So the idea is after 14,000, something must have happened, probably influence of the Berlin and Allerod warming event, which opens the landscape, maybe corridor, geographical corridor, reforestations of Europe from the south towards the north, and probably also hunter-gatherers follow, praise, and move north, replace the local populations of the Magdalena, which probably even moved north to the fringes without evidence of substantial admixture on this process from south to north expansion. And yes, this is the story about the Oberkassel cluster in Western Europe, but in Eastern Europe, you have another cluster, it's very distinct. So we call Sidelkino, or also previously known as Eastern hunter-gatherers. And from 14,000 to 8,000 years ago, in Western Europe, nothing happened from what we can see. There is no admixture whatsoever. So think about 6,000 years without really major admixture with population that are living side by side, or at least in the same continent without substantial contact. Up until 8,000 years ago, we see the first contact in mainland Europe with Eastern hunter-gatherers. Then right after bidirectional gene flow, up until 7,500 years ago, when we have the expansion of farming into Central Europe, the hunter-gatherer population retract themselves towards the fringes of Europe. And at that point, yes, we do see a lot of interaction within hunter-gatherer. We really see a lot of gene exchange between hunter-gatherer population, maybe because they were enhanced by the expansion of farming from the south. And yes, we also see some individual with this green component carrying farming ancestry. But think about up until, I always find very surprising, up until 5,200 years ago, we have in northern Germany some individual with 90% or 95% hunter-gatherer ancestry. And farmers arrived, 2,500 years before in Europe, right? But they were still this parallel society living side by side without, at least on the hunter-gatherer and forager society, without so much genetic exchange with the farming communities, contrary to what we see the other direction, where instead farmers really acquired a lot of hunter-gatherers ancestry through time during the Neolithic. So just to summarize here, I don't want to be too repetitive, but our Ignatian, so as a graphical summary, you have all these techno complexes, archaeological culture from 40,000 down to the Mesolithic. We kind of reconstructed this very complex kind of schematic representation of admixtures, continuity, replacement during these 35,000 years with multiple lineages, multiple ancestry, they mix and replace themselves. We kind of summarize here with this schematic, just briefly. We see that the pre-LGM, the Gravettian population were actually very distinct. They were not belonging to the same ancestry. We have at least two different genetic groups. We see that after the LGM, actually during the LGM, sorry, the Western Gravettian, this cluster, survives in Southwestern Europe. Instead, the Eastern Gravettian seemingly disappear. We don't find evidence of this ancestry anymore, nor in Italy, nor in Eastern Europe after the last glacial maximum. We have to say we don't have data from the Balkans so far, so we might find surprises, but this Eastern hunter-gatherer, Eastern Gravettian and Southern Gravettian disappear from the genetic record, whereas after the LGM, we observe a re-expansion of this Western Gravettian ancestry associated with Magdalena individuals from the southwest to the northeast. And at the same time, we see the spread of epigravettian-like ancestry, most likely from the Balkans. Again, we don't have data, but we speculate it's from the Balkans, entering into Europe. And then after 14,000, sorry, entering into Italy, and then after 14,000, entering in the Central Europe.
Minus 25.
Hello? Entering with the Oberkassel cluster, and living side by side with the Eastern hunter-gatherers for almost 6,000 years, up until 8,000 years ago. We have also evidence that this individual might be phenotypically different. I didn't present on that, but they might have different SNPs associated with pigmentation that made them phenotypically different, the Eastern and Western hunter-gatherers. And finally, after 8,000 years ago, you know the story, farming spreads with Neolithization, and this really pushes the hunter-gatherer population towards the fringes of Europe, and there is much more admixture between these groups, but also, of course, between farmers and hunter-gatherers. I would like to give you this example now about this study we just got actually accepted. So you haven't heard about that, but it's really related to the epigravettians. So I would like to spend a few more time to discuss about this very important specimens and site, Riparo Tagliente, where in the previous paper in last year, we published a good quality genome from an individual coming from this cave, or actually it's a rock shelter called Tagliente 2, okay? So this mandible is they named Tagliente 2, and we analyzed a tooth from this mandible, which is found actually outside of the rock shelter, okay? A few meters outside of the rock shelter, okay? Dated to 17,000 years ago. And so it's one of the oldest epigravettian genomes we have available from northern Italy. At the same time, we got access to Tagliente 1, okay? This is, we got access to a femur fragment from almost complete burial, which is found inside of the rock, was found inside of the rock shelter, okay? Tagliente 1, Tagliente 2. And so the question when we started this project is like, can we do a zoom-in study and see what is the relationship with these two hunter-gatherers, right? From the same site, are they related? Are they not? How do we find any local replacement, or what is their ancestry? And so, first of all, we perform radiocarbon dating. So there was already radiocarbon date done on both specimens, and this becomes interesting. You see what is going to happen now. So Tagliente 2 is 17,000 more or less. Tagliente 1 is 900 years younger. So the two dates are not statistically overlapping. I'm talking about the median age of the both dates. So we obtain this femur fragment from Tagliente 1. So we perform a new radiocarbon date, and the date became older, okay? Became older because also the same a newer approach was used compared to the first date of Tagliente 1, which was done, I think, in Oxford, but very long ago. So we said, okay, let's try to date it again. And indeed became older, became 450 years older, but still statistically not overlapping with the 2 sigma interval to the Tagliente 2. And so, of course, we said, okay, let's use this Tagliente 1 data also to generate isotope studies, okay? And again, we find that Tagliente 1 and Tagliente 2 are having different, distinctively different stable isotopic profile in terms of carbon and nitrogen, okay? Assuming that maybe one of these individuals had a different diet in life, and there had been already previously proposed evidence of consumption of fishes during this period, okay? So we said, okay, let's try to do genetic analysis, okay? And we first obtain mtDNA analysis, U2 haplogroups, very common in the Upper Paleolithic and the Italian Peninsula in particular. The same is the same haplogroup as Tagliente 2, and also the Y chromosome I2, very widely spread haplogroups. So really, this analysis is not conclusive. So we look at the nuclear genome of Tagliente 1, and we find that Tagliente 1 has incredibly high affinity with Tagliente 2 when compared to other Italian epigravettians, okay? You might say, okay, this is expected. They are from the same site, okay? Let's try to see what is the relationship, okay? And then we do another analysis called PMR, based on biological kinship, and we find that is the same individual. So yes, we were a bit disappointed because, of course, Tagliente 1, Tagliente 2, basically, we can really confirm it is the same individual or identical twins. Quite unlikely, but you know, we wanted to then revisit all the evidence and try to put evidence together, okay? And so, first of all, both skeletal remains, one found inside of the cave, one outside of the cave, have been morphologically identified as male individuals with an estimated age, surprise, surprise, of both between 25 and 30 years. Tagliente 1 is genetically identical to Tagliente 2. More parsimoniously, both humans remains belong to the same individuals. how do we explain the discrepancy in radiocarbon dating and in stable isotopes? Different stable isotopes might be explained by different diets in stages of life, right? In the adulthood and in the childhood, they might have access to different diets, maybe different amount of fish consumption in their diet. But the statistically non-overlapping radiocarbon dates, we suspect they are due to carbon-14 contamination. Because, and here comes the interesting part, the marine reservoir effect, or in this case, freshwater reservoir effect, cannot explain the signal. Actually, it would make the distinction of the two dates even larger, okay? So the correction would make the dates even further apart from each other, okay? And so we said, if we take the older dates as the real dates, because usually contamination tends to create younger dates, how much carbon-14 contamination would you need to get a shift in the radiocarbon date of between 450 and 950 years? And just here to give you an idea, I think people in the room know, but I think many others don't know, is enough, a very small amount of contamination, even at this time period, 17,000, to really shift substantially and make those dates not statistically overlapping. 0.7% or 1.4% is what we estimated. So we think that this is due to, of course, treatments of the bones, which we don't know because the taphonomic history post-excavation of the skeletons is not known. And the mandible most likely derives from disturbed context. And so the mandible maybe was even belonging to the human burial inside of the cave, which was brought out of the shelter because of disturbance during the medieval period, because there is evidence of some activities there. And so here really it's kind of a study where we see that if we put all the pieces of the puzzle together, we can really shed new light of the fossil record and really answer questions of bioarchaeological questions, which for a long time were not possible to be addressed. In fact, with this specimen were even labeled Tagliente 1 and Tagliente 2 because it was always being considered that they are indeed two different individuals. So now we group them together and we call them Tagliente 1/2 so that to avoid confusions. Okay, very last topic I want to briefly mention about, but since a lot of people in the room are working on similar topic, I think it would be nice the chance to interact with you more. We got financed since last year in Tübingen a campus which is funded by Leibniz Association. It's called Genomic Archaeology Campus Tübingen. It's a collaboration between Max Planck Institute in Tübingen, Senckenberg Institute in Tübingen, and university, of course. And the idea here to study the diachronic impacts of humans on cave ecosystems, okay? And why we chose caves, you know better than me, caves have sediments that derive from precise archaeological context, enabling a link with human behavior. So we have this kind of beautiful, long-lasting laboratory archive of DNA, which can study to see what is the impact of human, the presence of human, and how species change based on interaction of based on human presence. Is there coexistence? Is there replacement? Is there genetic diversity being lost or gained with human activity? And of course, you find DNA also in lake sediment. You find the ancient DNA also in permafrost. But in this case, we have a direct link with human behavior through archaeological surveys. So what we can find inside of archaeological caves, if you're lucky, if you're Nick Conard, you might find a Venus figurine or something other type of figurine. You find osseous material, stone tools if there is human activities, but the last majority are those bags and bags of sediment. So we try to get access to those and usually analyze, instead of skeletons at this point, which are extremely rare from this time period, Upper Paleolithic Mesolithic, and so forth, really focusing on western of caves. This is the evidence that this is possible, and actually many people in the room here have also contributed to this evidence that caves are fantastic repository of aDNA. There is also evidence that geoarchaeological blocks can be used actually to perform microsampling and obtain different elements of DNA, and Mareike is working hard actively on that. So in microfeatures that preserves DNA and possibly differential preservation. And then also, not to forget, this pivotal study, 2021, where they were obtaining to were able to show convincing evidence of a component of the Neanderthal gut microbiome in sediment associated with fecal material with Neanderthals occupation. So not only able to obtain the Neanderthal DNA, but you're even able to obtain elements of the gut microbiome of this population sampling the sediment. So we try to put all this together in integrated methods in Tübingen with working with colleagues in microbiology, bioinformatics, of course, archaeology, but also geosciences, to try to understand how and where DNA is preserved, and maybe just try to go beyond just mammalian presence and say, okay, mammals are present or not, but really see what else is present there. So we have kind of three components focusing on microbes, cave community, and demographic studies, more like, you know, replacement of Neanderthals by modern humans and so forth. Try to interlink these three components with 5 work packages. And without going too much in details, because I'm running out of time, but we can discuss about that later. We have a team, and just also want to mention that this is the GACT team, so scientific coordinator, assistant, but also 6 new hired members. And we have a new member of the team since last week, which is Hamilton, a liquid handling system. So we're really moving towards the parallelization of ancient DNA generation from extraction all the way down to index libraries. And so I think we can, I think many of us actually are trying to go in that direction to scaling up the output of generation of data, specifically when working on sediment DNA, where you have no really limitation on material. And I also mentioned that because we have project funds, obviously, to run the project, but maybe it would be the chance also to establish some contacts, some collaboration, because among the different pockets of money, there is one very interesting, is visiting researcher program. There is funds for researchers from other institutions to come to Tübingen for 3 months, get, of course, salary for 3 months, and then the trips covered. We had the first visitors at the very beginning, Nemanja Markovic. He, for example, worked on cave coprolites from hyenas from a cave in Serbia, and we find, for example, huge amount of diversity in those coprolites, not only the DNA of the host, in this case hyena DNA, a lot of mammalian DNA from most likely what the hyena ate, the Last Supper, but also microbial community associated with the gut microbiome of the hyena. So this opens, of course, a possibility and gives us a chance, I think, not only us, but also you in Vienna to really expand a lot in this topic, because you are just scratching the surface. And then this is the website, so you want to see what we are doing. We try to keep it as updated as possible, geogenearchtubingen.de. And then finally, I would like to thank my group in Tübingen, all the sponsors, and of course you, HEAS Institute, and particularly Martin and Ron for the invitation. Thanks a lot.